Okay, thanks, Connie. Can you hear me okay?
Yes.
Great. Yeah, so it's an honor to present along with Chris, my co-lead, on behalf of the Carcinogenomics project team. And Connie, if you can go next slide. Just before we begin mentioning Chris's disclaimer that his views do not represent EPA policy. And then also a big shout out to everyone that's involved in this project. We have a really nice team across industry, academia, and government, as you can see. Have made some really nice progress in the years leading up to 2023, but especially this year. So big shout out to everyone who has helped with that, and none of this would be possible without this large team effort. Next slide. All right, so you heard the talk from the error-corrected sequencing group earlier in the efforts to look at markers of clonal expansion using error-corrected sequencing. This project dovetails with those efforts with a similar goal of identifying early biomarkers of tumorigenesis. And so, whereas clonal expansion we expect to see on the timeline of probably months in animals, this team is focused on the molecular initiating events that induce tumorigenesis in the rodent with the hope that we can identify biomarkers within a matter of days that are predictive of potential outcome at 2 years. And so in 2022, this team published this overall strategy of combining the MIE biomarkers, you can go back real quick, with the error-corrected sequencing biomarkers as part of a weighted evidence approach, really to feed into the revision to the ICH S1B that came out, I believe, late last year, and providing this data to feed into that weighted evidence approach in an effort to first weed out bad actors before conducting 2-year CARSO studies and save those resources, but then also indicate when a 2-year study would not add value and being able to submit that as a potential waiver to regulatory agencies. Okay, so we can go next slide now. All right, so this slide just reiterates much of what I just said, but just to formally state the objective of this team is to drive international industry, regulatory, and academic understanding and acceptance of a weighted evidence approach using new genomic tools for practical application to in vivo shorter term rat studies to inform on tumor risk in the 2-year rat carso assay. Again, you heard from Patricia et al. For the ECS working group earlier. That group has formally spun out of this initial group and is now working somewhat on their own. And then so the Carcinogenomics team going forward is focusing on the MIE aspect of the project. So more specifically, what we're talking about then, the team is focused on liver, as that is the most common site of chemical-induced carcinogenesis in the rat. And we're focusing on multiple AOPs and molecular initiating events known to lead to tumors in the rat liver. So that includes AHR, CAR, PXR, PPAR, ER, chronic cytotoxicity, as well as DNA damage. Kind of pairing off Carol's prior talk from the in vitro and looking for similar markers in vivo. I think there's might be an animation on this slide that I didn't realize. Yeah, there it is. So it's just important to note that we're not flying blind here. This work is really leading off a number of publications that have been released by Chris's group and others, as well as our group at Merck, showing that there is promise to be able to identify transcriptional markers of these various transcription factors and MIEs, and that these can be predictive of outcome at 2 years. And so really the group for this team is to align on consensus of what those transcriptional signatures should be, as well as the levels of activation that are associated with tumorigenesis. Next slide. So we put together multiple working groups to enable this effort. That's shown here. There's a strategy and outreach team, which is responsible for communicating the outcome and strategy of the team and making sure we stay aligned um, with regulatory strategy as well as the ICH strategy as well. And then in order to do any of this work, we needed to first compile all the data that was needed. And so we had a data compilation team responsible for getting all the genomics data in line and analysis ready for analysis. And then also a mechanisms annotation team that went through all the compounds for which we have data to assign particular MIEs, as well as pull the outcome data from the 2-year CARCO studies. And all that work was really the focus of the first several years of the project, and then finally led to the gene signature development team getting off to the races really over the last year and a half. And I'll provide some updates where we are there. So what I'll do is I'll walk through some of the updates from each of these teams across the last year. But before I do that, just again outlining the timeline of the project. It initiated in 2019, following a proposal led by Frank Sester. The first couple of years, again, we're compiling the data and all the compound annotations, and then project's really getting fun the last year or so when we're able to dive into the data and start developing the various biomarkers. And then Chris is going to end talking about some of the in vivo studies we're doing to further bolster this data set, looking at knockout rats for each of the MIEs. Next slide. So this slide outlines the work that the strategy and outreach team has done. So we published a manuscript in 2022, but then in 2023 presented a poster at SOT. We're submitting another poster for next year to update on the progress. And we're also planning to present an SOT webinar in February. And importantly shown there in the picture is, as we all know, Connie. But big shout out to Connie, because her project management skills of this project have been critical for moving things forward, nagging us to deliver results. And we really wouldn't be where we are without her. So thank you, Connie. Next slide, if you don't mind. All right, real quick, the data compilation team. This effort mostly completed in 2021, so we presented this slide before. But what that team did is pulled together 6 different rat liver transcription data sets, mostly from Affymetrix, but then also some RNA-seq data as well. That data set covers 370 unique compounds that we'll use to derive the signatures and test prediction of the tumor outcome from among those compounds. The team pre-processed and cleaned all the data and then made it available for the various teams to dive in and start doing analysis. All the data is hosted on SEBS. I should have added to this slide that this year, Scott Auerbach's group at NIEHS helped put that data into a SQL database, and that's made it easier for us to share and have the data analysis ready. Next slide. So the compound classification team, not the funnest job, but definitely a lot of work and critical for any of this. So huge acknowledgement to the progress of this team. Last year, the team was focused mostly on annotating the various MIEs. So going into the literature for data that supports whether a particular compound activates the AHR, CAR, PXR, etc. And all that data has been rolled up into the database. And then this year, focusing on pulling in all the 2-year CARCO bioassay data that we have available for these compounds. So that data is mostly pulled in, and we're currently structuring all that data into a machine readable format. And so hopefully to finish that up in the not too distant future. Next slide. Right, so all that work goes into being able to derive the gene signatures. And so important to point out that there's 2 parts of this. The first part where we are now is to derive gene sets that are indicative of activation of each MIE. And then part 2 will be to look at the signature responses, the quantitative responses from each of those gene sets, and see how it compares to tumor risk in order to set levels of activation that are associated with tumor risk. So in deriving the gene sets, we're taking several different approaches to ensure that we're both sensitive and specific. So straightforward approach that you'll often see on more limited data sets is you take several compounds of a particular MIE, treat animals, and look for the consistent changes by those compounds. And that'll identify genes that are sensitive for that MIE, but not necessarily give you genes that are specific for that MIE. So one of the benefits of this large data set we've compiled is we can look at the effects of these genes across a very diverse set of chemistry, agonists of different receptors, and confirm that the gene sets that we're deriving are also specific for the specific MIE. And then Chris will also talk about work we're doing to ensure that we're targeting the genes that are most proximal to each transcription factor for each MIE, specifically using the knockout animals to confirm not only is the gene downstream of the transcription factor, but also using approaches like ChIP-seq to show that it's directly targeted by the transcription factor. Okay, next slide. That's good. Yeah, so the approach we've taken, we didn't want to script the gene set development approach too much upfront. And the rationale here is that there's any number of different analytical approaches one can use to look for differentially expressed genes or potential biomarkers of these MIEs. And the idea is that a good biomarker should be represented across any one of those approaches, right? So a robust biomarker should be obvious across, regardless of your analytical approach. And so what we did is really release the data to the experts within the team. And so they're among the groups participating in the project. There's a number of bioinformatics teams across groups, and we've been allowing groups, handing the data to those groups and allowing them to essentially play with the data, take their best shot at identifying gene signatures and to see what they come up with. So we asked that in order to be prepared for this meeting, that the teams would bring to us their current favorite lists of genes from their analyses to provide them to us by, I believe it was Monday of this week. And then we quickly looked through the consensus of those gene sets to see what the results were looking like. So we so far have gene sets from 4 different groups, from Scott's team, who used a linear support vector classification approach, from Heather and Ashok at Takeda, using indicator species analysis, from our group at Merck, where we took a similar approach to our previous publication based on RNA-seq, but then just applied those analytics to this data set on the Affymetrix data. And so we supplied those gene sets. And then Cody and Elias at Pfizer took a random forest approach. So if you go to the next slide, this is some late breaking data on how these results are looking. And so what I'm showing here, the numbers in this table indicate the number of each of those 4 approaches. So from the 4 different teams had that gene within their signature sets. And results are really encouraging so far. So among the 4 sets of signatures that we got from the 4 teams for each of the MIEs, there were common genes identified for AHR, PPAR, CAR-PXR, ER, as well as genotoxicity. And so that's what I'm showing in the table, highlighted by MIE. And if you look at the genes in the that are listed there, the really encouraging part is that these are not unknown genes. We're picking up, canonical targets of these transcription factors in these analyses. So really encouraging results so far. One fly in the ointment is there are not yet any common genes identified across groups for cytotoxicity. There's a couple of reasons for this. One is we're classifying cytotox a little bit differently across groups, but then more importantly, we know there's a strong relationship between cytotoxicity and genotoxicity responses in the liver, and so it's hard to deconvolute those signals. We do know from our prior analysis, though, that if you look across time, you can start to resolve those signals. And so this is something the team's just going to have to dive down deep. We expected this to be a confound in sort of these early approaches, and it's something we'll be following up on. So stay tuned. This is, again, late-breaking data. This list will probably change a bit, but really encouraging so far. Tomorrow, we'll be having a working group where each of the teams is going to present on their specific approaches, and we'll talk more about the details and strategy as far as reaching consensus on the sets going forward. So if available, we welcome you to join those discussions. I have one more slide just to talk about the next steps from the informatics approaches, and that'll be to identify the levels of activation of each of these MIEs that are associated with tumor risk. And we know that it's possible to activate these MIEs without an issue, right? Every time you have a plate of broccoli, you're activating AHR in your liver. There's a seminal paper from Mark Feldman et al. Showing that a lot of pharmaceuticals have some low level of AHR activation. And so it's really the sustained hyperactivation of these MIEs that's the issue. And Chris has done some really nice work showing that indeed he could identify thresholds for each of these markers that are associated or not associated with outcome at 2 years. And so that's really going to be the next step of this working group, is taking these consensus final signatures and deriving those quantitative thresholds. So Chris, with that, I will switch it to you.
Okay, thanks, Keith. That was a nice overview of the project. So what I'll do is I'll talk briefly about a study that's been funded by the eSTAR and HESI to help support this project, where we're going to be generating gene expression profiles and biomarkers from wild type and knockout mouse comparisons. So next slide, please. So we recognize that a chemical centric approach to build and test the biomarkers could have some complications. And I think we've already seen that in the spreadsheet that Keith showed, that there is a fair amount of overlap in the genes that are coming up as being the biomarker genes for the different MIEs. And that's just because these chemicals are dirty and they're activating multiple chemicals. And so what we were thinking was that, last year when we proposed this project, that we could help to identify those genes which are specifically regulated by the different transcription factors that we are going to be predicting. And also this was fortuitous in that Envigo was offering the knockouts to do the study. So if you go on to the next slide. So no, go back one. Yeah. So
no, these ones have a timer on it, I think. Sorry, I'll try and get rid of it, Chris.
Maybe I should talk faster. So we're going to identify genes using this wild type versus knockout comparisons, and we feel that this is going to give us an unequivocal assessment of the factor dependency. Now, when we generate these genes from these comparisons, we could use them two different ways. We could either validate the genes that have been identified using the chemical-centric approach, or we could build the biomarkers de novo and then test them on the microarray data that's already annotated for the factor activation. And a lot of this was supported by past studies. And if we look at this next slide, there are three different examples of studies in which capitalize on the availability of microarray data from wild type and different transcription factor knockout mice. And here we're looking at the construction of biomarkers for PPR alpha, for CAR, and for AHR. In each one of these cases, we're looking at, or the data that was available was AFI data that came from wild type or the knockout mice treated with 3 different chemicals. And so there are a lot of ways to identify these genes and to build the biomarker. But in these cases, there were certain rules that were followed, and they built the biomarker. Most of the genes were those that were upregulated. But the important point is actually on the next slide, is once you use them to make predictions, then the predictive accuracies of these biomarkers were excellent. In fact, they range from 94% up to 98%. And here are examples of 6 different biomarkers that have been constructed. So there's a lot of historical precedence that this approach, using the knockout approach, is going to work. So next slide. So we had a lot of discussion about how we were going to use the resources from HESI to do the best study that we can that helps supports and addresses the questions that we think are important in constructing these biomarkers. So the knockout study consists of 4-day rat studies where we're looking at comparisons between wild type and the knockouts for AHR, CAR, PXR, PPR alpha, P53, and the ESR1 or estrogen receptor alpha. And all of these studies are being conducted in Udayan Apte's lab at Kansas University Medical Center. And what we decided on was that each of the studies would look at 3 reference activating compounds, and that the wild type rats would have be treated with 2 doses, both a low dose, which was below the tumorigenic threshold, and then a high dose, which was definitely tumorigenic, whereas the knockout rats would be treated with just the high dose. The livers would be then sent to Merck for processing and RNA-seq analysis. And we've been having discussions about capitalizing on the availability of tissues from this study that could help to address the factor dependency on or in other tissues. So if there is interest in obtaining some of these tissues by others who are listening, then contact Connie and we'll get you involved in these discussions. Okay, next slide, please. And so here is the timeline. I'm happy to report that the first block of this study, and that is the study looking at wild type and PPR alpha knockout rats, has now been completed. Udayan and his group are now soon to expose the ESR1 knockouts, which will be conducted next month. And it's fortuitous that the only lab that we could find that had the ESR1 knockouts was actually at the institute where Udayan is, at KUMC. Later in 2024, he'll be working with the P53 knockouts, and then AHR, and then in April, CAR and PXR. So by summer, we hope to have the complete set of livers, which could then be processed by Merck and get some interesting data. Next slide. And this last slide just summarizes the project in that we'll be, you can go back one, and then that we will be looking at comparisons between the wild type and the knockouts, 3 different chemicals using, and in the transcriptomics will be conducted using RNA-seq. We'll use a number of computational approaches to generate sets of genes that we know are completely or partially regulated by the transcription factor. And we hope to then generate 6 non-overlapping gene sets that could be used, as I said, predictive biomarkers or to validate the chemical-centric approaches. And the first set of results we hope to obtain early next year. And that is it.